All right. Hi, everybody. Welcome to Asynchronous Human Phys. So today we're going to cover lung capacities, and then we're also going to kind of look at how we regulate rates of respiration, things like that. That's going to go back again to lab 1, where we looked at how your body kind of keeps things regulated at its set point, wants to keep things pretty average for the most part. Now, we'll get to see some cool examples where you don't want that. For example, like exercise, where you need to be, breathing a little more. But before we get to regulation, we get to go over capacities, volumes, and kind of just the nature of why your lungs do what they do and how that relates to a lot of stuff. So some cool examples exist here. First of all, exercise. Everybody in exercise phys here, this is kind of your time to shine. Given, kind of the differentiations between different patients or different athletes, for example, that can, have a higher capacity, you got to find out why in a lot of cases. These are the basic rules in some cases why. Some people have a different capacity for, better lung volume and things like that. Other disease examples include something like cystic fibrosis, where somebody does not produce the necessary molecule or the necessarily mucus in their lungs to basically have it like smooth enough to operate the same way, things like that. That's a genetic disease. Another good one is altitude. So if you're like me from Colorado and you were born at like 6,000 feet or something, we have a greater lung capacity. Somebody though, let's say from Peru, who's born at 10,000 feet, has an even higher VO2 max. I don't think we directly see VO2 max in this, but again, it's kind of a cool example to keep considering as we go through this. Let's take a look, and you'll also see that these two things are not always associated to be the same, but the whole idea of blood doping or altitude training and stuff, that's something to keep in mind as we look through all these examples as well. Kind of the other, we'll see some other stuff. I know I'm going to spell this wrong. Emphysema. Oh no, I think I ruined it. But we'll look at restrictive lung diseases, things like that. Me as a cancer person too, one of the other things that is often associated with cancer is kind of the end stage of cancer, which is metastasis. That's where the tumor, wherever it's from, starts shedding pieces of itself. They go and find another place to grow. Big problem is that lung tissue is a great place for tumor cells from a lot of different other tissues to grow. New little tumors grow in the lungs. They restrict the volume in the airways. Eventually, you can't overcome that decrease in volume and decrease in stability, and the patient dies. So if you ever want to talk about some more tumor stuff with me, I'm always around. That's kind of my field. So if you're interested, let me know. Otherwise, let's kind of move on here. So look at this, physics and chemistry, Boyle's law of gases. Oh no, of course it was going to catch you eventually, right? So good thing here is that this is pretty simple. If you have a high volume, you got a low pressure. Intuitively, if I have a big space, there's not a lot of, of the same molecules, let's call them air molecules, oxygen molecules, whatever. If I have a big space, they're not really pushing on that area very much. This is very similar again to the blood pressure, right? But now we're just talking about air. If you have a small volume though, with the same number of molecules in this small volume, you are now having a high pressure. There's more of them pushing against their volume in this case. So keep that in mind and know that this inverse relationship is going to, as you go down in pressure, up in volume, same thing's going to happen if you reverse these two things right here, a lower volume, a lower pressure. Okay, Penny goes into this well right here. And again, like I've reminded you in all these, the notebook is where all the fair game comes from, okay? Anything that's on this lab exam is in this small area right here, and as well as the data that we're going to see today. So those two things are what's important to keep in mind for these labs. Okay, other thing to keep in mind here is that if we're looking at airflow, you got two factors that you can look at. One is pressure, one is resistance. This is again kind of a semi-physics equation type thing, but at the end of the day, resistance is something to keep track of because this is where you do have those restrictive lung diseases and issues in that regard. Okay, so if we move on right here, and it actually starts getting in right to it. So if you have high resistance, you have a lower airflow. So see, if you have a high number down here in this resistance right here, you're gonna have a lower flow right there in that case, as far as air goes. So anytime you have one of these issues, for example, you will have a decrease in airflow because all of these increase resistance. In the case of COVID, COVID is attaching to the cells in your lungs primarily. Damage can come from the immune system reacting in other places, which is the nature of the disease and why it finds an area of weakness. However, its main target is the lungs because these are the cells that it's multiplying in. If you have one of these conditions, or if you have patients that have these, then their lungs are already have kind of a higher resistance. And if you keep damaging some of the lung cells, that resistance gets higher and higher and higher. One of my friends has asthma. Actually, I've had 2 friends in Colorado get COVID. Both of them have asthma, and they both got really knocked down by it because they already deal with kind of a handicap in that regard. Okay. So some of this is detail right here that I'm not going to, belabor for you, but you have space in here, and this is where the physics and the pressure and the volume are going to come in. You have this chest cavity and the expansion and de-expansion. What's the word I'm losing here? Suction. Yeah, there we go. That up and down of the lungs is very important as far as a regulation point of view. So as you inspire, for example, pressure is going to become negative, and this allows the lungs to collapse. If you're breathing out in this case, and the pressure becomes less negative. That's the back and forth, and it's all a control of pressure. Okay, so this is why if you puncture the lung, for example, let's say a rib goes through it or something in a car accident, you've lost all that control of pressure all of a sudden, and that's why puncturing a lung or something like that is so devastating. Now, it depends on how big of a puncture you've got. If it's one that's going to let all that pressure loose, all of a sudden now you can't fill up your lungs anymore because they're just like losing pressure as the air goes out. Okay, let's go over here. So if I actually just wanted to zoom in right here and just kind of go through a quick like run through right here. As the intercostal muscles contract, causing the thoracic space, in this case, thoracic. This is going to cause the thoracic cavity to expand. This is going to lower pressure. When you lower pressure, the lungs themselves expand. When the lungs expand, that higher volume is going to lower the pressure, in this case, again, in the alveoli, I should point out. And these are little like pockets, little bubbles in the lung that are like kind of filling and like going up and down. And what this happens is that lower pressure in the lungs is going to equal that the atmospheric pressure, which is just everything outside that we're breathing in, is now allowed in. Okay. So your lungs aren't so much sucking air in as we're reducing the pressure in the lungs, and naturally, just remember with empathy, remember with like kind of with diffusion where things equal out. If we lower the pressure, now the pressure around the environment can equalize that and come in, and then the process breathing can kind of continue in that case. Okay, so in this case, does a good job kind of highlighting some good examples here when you're passive at rest, like what does this look like versus when you're exercising. So that's a good example to think for yourself. What are the changes in all these scenarios that are going to happen, let's say, if I am jogging or if I am sprinting? And that's going to be something to apply all over the place. This is also a really good table here. This is something, again, if you want to try the retrieval practice stuff, this like arrow, 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 and like set of things that happen over and over and over again. That's the first thing to practice with processes. That's how I learn processes is this happens, arrow, this happens, arrow. Then you can expand on them later. Okay. Oh, yeah, this is a good one. Surfactant. So this is kind of it's a bit of, yeah, it kind of reduces tension in the alveoli as they're expanding back and forth. And that expansion back and forth, you need like a smooth liquid like layer to slide back and forth basically. In this case, like to facilitate this alveoli collapse, it keeps them from collapsing in this case because it is transitioning, like those cells can move smoothly around each other. So there's a good example right here where basically a big deal with premature babies is that if they don't have this fluid, their lungs are not, they're kind of rigid. They're not firing on their own yet. And in that case, that's where you have to, I I don't know if intubate's the right word here, but you do have to help the premature infant for quite a while with its own breathing while those cells develop the liquid. Okay. So let's go into some data right here. This is a good chance to kind of practice and see what we're actually going to be using when we do the data. So in this case, here's our nice lung volume, okay? All of these right here, these are normal. These are just normal breaths, okay? This is something called tidal volume. This is when you're just sitting here, just breathing back and forth, everything's fine. That's your tidal volume. When asked though, let's say somebody, health professional asks you, take in as big of a breath as you can, right? You're gonna reach as high and as big as you can in your lungs. This is your reserve. This is your, I wanna say this is your VO2 max. This is again where somebody from altitude theoretically is gonna have a higher VO2 max. Swimmers, in a lot of cases, have some of the highest VO2 maxes on the planet. So like Phelps, for example, he has a massive VO2 max because you have to take in as much air as possible in as few breaths as possible to be a good swimmer. So putting these two together is what you have called your inspiratory capacity. This is how much you can take in. Now, in this case, after you've taken in this massive breath, you're going to have to basically dump it. This is your expiration reserve volume in this case. This is how far you can push out. And if you ever tried that, it's actually a lot easier, as you can tell, to bring in a lot than to push a lot out. Pushing a lot out actually kind of hurts, right? It's like, holy crap, I can't breathe anymore. This is the point at which your lungs can fully expel as much as they can. And they're not as well built actually to do this. So that's where you have residual volume. This is where you don't actually ever want it to go to zero. Your lungs are actually built to not let you expel enough out. If you do, you lose all kinds of pressure, things like that. Things get really bad. And again, that's where if you puncture a lung or something, you start going into this area and you don't really want that. Okay, cool stuff. And again, this is actually a very good diagnostic measure right here, vital capacity. This is what's your up and down? How much can you handle? This is a huge thing for fitness. PT, OT, how far can you push your clients in this case? And this is a big measure because remember, if you're PT or OT, you got to customize it for everybody that you're working for. Not everybody's built the same. Not everybody's had the same lifestyle, things like that. Like my poor dad, if he had to do this, I don't think he has a very good VO2 max because even though he grew up in Colorado, because he still lives like he's 25, so it's a bit of an issue. Okay, so we have our good measure. Everything's I hope I kind of went through everything in here already, just visually up there before. So again, this is a good chance to try going through something like this on your own and drawing it on your own, pointing everything out, what it means physiologically, what are good examples for each. The areas that you don't get, fill all your gaps with the text. The text is everything you need. Remember, this is the only place we get fair game questions for the exam. And remember, this exam is going to be online, so it's open note. That's okay. Don't worry. At least that's how I'm designing mine, because it's weird that it wouldn't be, right? Okay. In here, I'm looking through this. Yeah, I think we've covered everything except FEV. Okay. So the volume test is FEV. This is basically your diagnostic tool for how much capacity do you have in your lungs. In this case, the test itself is going to be how much can you expel in 1 second. So it's going to be really fast, right? And how much can you huff out in that amount of time? Stronger lungs typically are going to be able to huff out a lot more in that case, right? So then VC, remember, is vital capacity. So this is your entire equation right here. This is the entire volume that you can huff out. How much of it and how much of it can you do in 1 second? So an athlete, for example, is going to have a higher amount that they can push out in 1 second and back in, because that's naturally what you're going to want to be doing in athletics. Somebody who's not doing so hot, for example, and let's call this what it is, somebody who maybe is more susceptible to COVID, is going to have a lower amount here because their lungs are not built to exhale as quickly and they're not as efficient in this case for whatever reason. And it's not somebody's fault if they don't have that, right? There's a lot of reasons behind, why you have, the lung capacity that you do. So, okay. Great examples here about the differences between obstructive, lung disease right here. This is when you have a high resistance. High resistance, remember, slower airflow. I think it was like, here, if we call airflow equals, I know resistance was right here, and then change in pressure. If you have resistance, whatever that is, it's going to cause less airflow because higher number here equals lower number here. Okay. My arrows really aren't on their game right now. I'll try to improve as we go. Restrictive is a little different. This is just straight up less air in or out of the lungs. This is maybe more of a volume issue, right? This is going to reduce your VC. So one of these is going to reduce efficiency, and that's obstructive. One of these is going to reduce total volume in that case, and that's restrictive in that case. So try and think to yourself what diseases, what conditions seem to fit with either of these, and then think why. Remember that thinking why something happens is really how you learn stuff. It's not flashcards, May not be the same thing for anatomy, but hey, it's coming from somebody who pretty much dropped anatomy, so not my style. All right, as we go through, we are going to be measuring volumes. This is unfortunate that we can't actually do this in person because it's actually kind of fun. And I usually, whenever we do this, when I've taught this in phys before, I always challenge the class to see who has a better VO2 max than me because I'm from Colorado. And again, it was the only one that's ever beat me was one of my swimmers last year. And I was like, holy crap, he beat me by like 2 times. It's crazy. Okay. What you're going to be looking for is this data. Okay. Now remember, tidal volumes, we covered these. This is just you sitting down, taking breaths, okay? This is nothing special. That is there's just pixels there. It's so sad. Okay. I think there's a better picture coming up here. So looking through here, I can read it on mine. Right second liters. Oh yeah, this is just volume basically. This is in liters of volume basically. And then I'm pretty sure this is volume per second. So this is how quickly up and down you can go versus this is just your total up and down. So with your deep breath, this is where you see that massive like upwards and then hold and then out. In this case, we're not seeing that big expulsion here in this because you're just going back between both of these. You really see that heavy expulsion in the amount that you're expelling per second right there. So that's something to note in this graph, I'd say. Okay, let's take a look here. So one thing that we want to graph to start is, in this case, I'm pretty sure, yeah, what does it want you to do? Oh, it just wants you to zoom in on this. So the data that you'll get will reflect this, and you'll be able to zoom in on something like this. What you want is 2 tidal volumes, 1 vital capacity, and 1 small tidal volume after. So the 2 tidal volumes that you're starting with, those are going to be like, what does a normal breath look like for this patient? What's their typical up and down capacity? Then you're going to have them go up, hold it, and then they go down. Now, from this, you're going to be able to get vital capacity. Because remember, VC is the whole thing. So vital capacity is going to be from this down to this top. See, from here, right there to right there. What that is the furthest inspiration you can go and the furthest expiration that you can go. That's VC. Remember that FEV is only how much you can expel in 1 second, basically. How quickly can you expel? How efficient can you expel? Very different than just the whole thing. Okay. Let's keep going down here and get specific. So first measurement that you're going to take is 1 tidal volume. That is full inspiration, full expiration, but just passively sitting. This is going to be a pretty small amount, right? This is your typical, just breathing. Now, if this was really high, what kind of disease would that indicate? So that's a thought question for you. It's not a good thing for sure, because if somebody's really struggling like, like that, it's not a good deal. And it's a good sign that you probably need to get them to an emergency room or something. Okay, so this is tidal, just typical breathing, pretty normal. Okay, now let's look at VC. What can your patient do? What's the best they've got? And remember, what I should highlight too is you're going to be using the journal icons here. Remember how you can like do the journal icon, scroll across? That's what you're doing here. Okay, same thing you're going to do here. VC is full inspiration. Okay, you're going to hold that breath and then it's going to collapse. You push it out as far as you can. Full expiration. And you're going to get that difference in volume. And that's your VC. That's your massive amount that you can actually, that's your vital capacity that you can actually take in and take out in your lungs. Again, going to be bigger in somebody from altitude or somebody who's like greater fitness. Not necessarily the same thing. I cannot take credit for that, unfortunately. But if any of you are from altitude and you've been walking out like on a mountain at like 8,000 feet and all your friends are more like, they're more fit than you and all that, that's good. But once you get up to altitude people take over and it's the best thing ever. It's pretty much my only talent. That's really all I got on some of my friends. Okay, so you measured VC. Remember, VC is here to here, top to bottom. But what we need for that FEV, and remember, this is going to be more of an efficiency measure, not a total measure. We need how much from the beginning of the expiration to 1 second later, how much was expired in that case, how much was pushed out. And you need just the 1 second for the measurement to work in this case. So using all that and what we know, I think pretty sure the questions actually stay the same, right? Like you just pretend that your data in these questions is one of the data that you chose. So remember, for this exercise, you pick one set of data that I submitted for you. Okay, go back. I think we'll go to 14. Oh wait, here I'm gonna do. Yeah, I tried to take a picture of Scout and we did the fisheye thing to her. So look at my hideous Pomeranian, sorry. Just had to plug that. Break time right between the two. All right, 14. Now we're gonna look at how you regulate this. How do we control the usually tidal up and down? Remember, tidal is the resting amount. This is really what we're controlling most of the time when we're just sitting, walking, doing stuff, right? So in this case, again, we're definitely going to be remembering, definitions from lab 1, feed forward, negative feedback, set points that keep you on average, things like that. Okay. So the best way, well, the way that you actually control things is, again, you have sensors, chemoreceptors that are going to tell basically how much O2 do you have in your blood, how much CO2 do you have in your blood, and in a lot of cases, how acidic is your blood, because the acidity of your blood is going to be directly related to the amount of CO2 in it. So let's take a look why. So let me find where are my little sensors? Yeah, there we go. Okay, so you have chemoreceptors. These are going to detect the 3 things we just talked about. They're going to tell your medulla in your brain, your integrator, hey, there's too much CO2. Oh, hey, there's not enough CO2. What are we going to do about that? They're going to send signals to your respiratory muscles, effectors. And again, remember, a lot of this is automatic, right? Like we can control when we breathe, right? But a lot of the regulation, the day-to-day stuff, we don't really put a lot of direct thought into it, right? Like, do you remember how aggressively you breathed yesterday? Not entirely, right? So in this case, the 2 more important things to look for are what's your level of CO2 and what's your level of hydrogen ions. The reason is that there's an enzyme that catalyzes CO2 and water into hydrogen ions in this case. Bicarbonate too, but that's kind of just a different, if you're more of a chemistry person, focus on it. If you're not a chemistry person, just relax and let's mainly take a look at these 2 things. So constantly, as your body has a lot of CO2 in it, it's getting metabolized into these little H ions. What the H ions do is when you have a lot of these, what that means is your body is acidic. Your blood is acidic in that case. So a lot of these little H pluses, these actually lower pH because, and remember, a low pH means that it's acidic, and that is brought on by lots of little hydrogen ions. So again, if you're not a chemistry person, relax, it's okay. We're just gonna like look at some of these relationships and see basically how we can use the basic chemistry to assign how much CO2 versus O2 you have in your body. Okay. So in this case, first thing we're gonna look at right here, if too much CO2 is present, more of these acidic ions are gonna be formed. Blood pH becomes lower and your body's gonna react to that. Similarly, if not enough CO2 is present, these H ions right here are going to combine with bicarbonate. They're actually going to re-up into CO2. And that's kind of a cool, interesting reaction if you are a chemistry person, but again, sorry. But in this case, if you have less of these little ions, that means your pH is going to rise. Because remember, the little H ions, they're the sign of acidity, which is a low pH. So less of these ions means a higher pH. That's called a basic pH. That's above 7 on the scale. Everybody actually did take chemistry and phys, so this should actually be super fun for everybody. I totally forgot that. Okay. The regulation of blood pH is your direct tie to how much CO2 levels are present, basically. These chemoreceptors are the ones that can detect how much acidity is around, basically. Okay. So in this case, aortic body, sorry, I just always forget. The little p is basically saying this is what we're using to detect the CO2 in that case. Why don't I zoom in here a little bit? Okay. So what happens when your body has too much CO2? We covered this right here. It's going to get acidic in that case, and your body's going to expel a lot of that CO2. So acidity equals expel the CO2 right now as much as you can. So why don't I actually, oh shoot, I don't have the, yeah, let's just go here, just a sec. I don't have room otherwise. There we go, okay. So in this case, oh no, don't do that. I don't know why it's writing that, sorry. It's like my palm. Okay, if you have high CO2 in your body, so you're not expelling enough CO2, you actually probably need to like up your breaths out, I believe. So if you have too much CO2, this is going to equal a lot of these little H ions. This means that you have a lower pH, and that's going to be detected by those chemoreceptors, so CR. Again, same inverse relationship is going to show up, and you're going to have a high pH, in this case, a basic pH, because you have a low amount of these sodium ions. Again, it's going to be detected by your chemoreceptors, and they react. They're the sensors. They tell your integrator in your brain, your medulla, to start increasing or decreasing the respiration. Okay. So in this case, we are going to kind of simulate some of these scenarios. Oh no, poor Scout. Such a terrible photo. I have such good ones ever. All right. In this case, we're going to actually take a look at hyperventilation data. So if you breathe in and out way too much, this is what's going to happen here. Okay. Since you're going to eliminate too much of this CO2 in this case, low CO2 equals low pH. Oh, sorry. See, look at me, this is hard. Low CO2 means low hydrogen ions, which makes a higher pH. Okay. Yep, we saw that. Increasing blood pH. This causes the chemoreceptors to signal control center to reduce the respiratory rate. Okay, so decreased ventilation will bring CO2 back to normal. Okay. In this case, we're inducing this bad change with hyperventilation, and we're basically decreasing the amount of CO2 that we have in the blood. And that's going to tell our body, okay, we need to reverse this and get this back to normal. Because remember, if we have a low CO2, we have a lot of corrective mechanisms to get us back to the set point. And that's the point of these systems, is to always get back to the set point. Okay. Two other examples, both of these are actually going to do the same thing. You can think about why, but for the most part, I can kind of just guide you through this. Rebreathing or breathing through a tube is going to increase dead space. You're essentially, what's happening in both these scenarios is you're going to be breathing in stale or used air. Used air has been converted a lot from oxygen, mainly to CO2. So if you breathe in used air, you're going to be breathing in quite a bit of CO2, more than normal, right? So higher CO2 is going to be higher hydrogen ions, which is going to lower pH in this case. And that equals kind of an acidic regulation in the blood. That acidity is going to trigger your chemoreceptors in this case to increase ventilation to get rid of all this CO2 that you've accumulated. Okay. So that's the basic principle. And here, I should finish this out, yeah. Okay. Painstakingly writing everything out like an old professor, sorry. So when you're rebreathing air, we're basically just simulating, you're getting way too much CO2 in because most typical air, just like in a normal room or outside, is pretty oxygen rich. But if you're breathing into a bag over and over again, you're converting all that oxygen into other stuff, and you're running out of that element. And the CO2 that we make as a byproduct is replacing it, and we're still breathing that in. So in a lot of cases, this is the amount of CO2 that you keep inside you is kind of actually a big deal about stress. Because if you are stressed, a lot of the time your exhale is not powerful enough, and you're actually leaving in quite a bit of CO2, and that causes your blood acidity to kind of go up and your ventilation to go up to recompensate. And so you're just kind of stressed all the time, breathing chronically a lot higher and higher and higher. And that's kind of an unfortunate thing, that stress is also going to increase the heart rate, hypertension, all kinds of bad stuff. So everybody chill, right? Easier said than done, unfortunately. So last thing for all my exercise biz, exertion. I don't think we're actually going to do an exertion exercise in this. The weird thing is that the CO2 levels actually don't really change with exertion. You're actually just, your gases and the gases that you do have, they are going to change in this case. So when your body needs more O2, when you're exercising, it switches basically the pedals that it's hitting so that more O2 is getting brought in instead of CO2. So additional O2 needs, you need to eliminate all the extra CO2 that you're breathing out, let's say while you're sprinting, and you definitely need more O2. This is going to cause, the respiration increase in exercise is actually feed forward, because as you increase respiration, you're expelling more CO2 and you're increasing your O2. So this is pretty intuitive, right? During exercise, you need to increase respiration because you need less of this and more of this. And pretty simple, right? In any case, still try and apply that to any situation that maybe is more specific to you. A sport you played, a patient scenario, things like that. I'm pretty sure this data is actually a little more like self-explanatory. I think we don't have to like kind of cover it quite as much, but in this case, you kind of see like in this case, that's your normal regulation up and down as far as respiration. When you do begin the hyperventilation, obviously, if you're going back and forth, there's a much like denser rate of the hyperventilation in this case, but you do have a recovery period that has some differences in it. And that's what you're kind of going to be looking for. When you start breathing in this kind of this dead air in this case, let me find this. Oh, it doesn't go in, does it? When you breathe in the dead air, again, you're going to see some differences in the data. What is the difference in the data? Actually, I'm kind of more curious now. All right, blah, 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 blah. Typical wave. Oh, Yeah, it looks like your waves are just going to be a little different in this case. Is that what it's saying? Measure a single wave. B, okay. Yeah, 14.3, right? Okay, see, this is the hyperventilation one. I was like, what the heck? Oh, okay, so there's no picture of B and C, I don't believe. Oh, okay, shoot, sorry. I was looking for the wrong thing. Okay, so you're all good. Typical questions. Like I said, I'll be around the first hour of each lab at the least. I'll probably have the computer on longer than that, but show up in that first hour. I'll probably break everybody into their typical breakout rooms, or you can tell me if you wanted to go into a different room with people. Because again, like I said, I completely freedomed up the groups here just to maximize scheduling stuff. You can turn in the assignment individually with your normal group, with a different group. You just got to put on the assignment like who you did it with, okay? Because as we deal with the reemergence of COVID, I'm kind of the philosophy of keeping things as open and as easy as possible to make that transition. Maybe for the kidney lab, we would do a live lab because we'd be, used to it again. But at the same time, it's only for one, so we'll kind of see. All right, cool. Let me know if you want any other cool or hopefully helpful examples with this. And like I said, the lab notebook text, that's where you're going to find all your points for this exam. And like I've talked to a lot of you, I do want everybody to do really well in this exam. So let me know how I can help, and we should be good and go from there. Thanks a bunch. All right. And